And then we're ready for the final talk of today, called On the De Novo Evolution of Mutualism in Symbulation. Please go ahead.
Thanks. Okay, I'll try to go fast so that we don't miss our boat. So I'm going to be talking today about some work that I did in collaboration with my undergraduate student, Connor Croft, and incoming PhD student to my lab, Kai Johnson, and my collaborator at Carleton, Anya Bostonar. Okay, so we wanted to study mutualism in the context of endosymbiosis. Endosymbiosis is basically situations where there is one organism, the endosymbiont, living inside another, which we call the host. And when we have these situations, there's going to be some interaction between these two partners, and those interactions fall along a spectrum. So they could be mutualistic, where both partners are benefiting. They could be commensalistic, where the host is kind of unaffected, and the endosymbiont, we usually say, is benefiting at a minimum because it has a place to live. Or they could be fully parasitic, where the endosymbiont is benefiting at the cost of the host. And often in these circumstances, the host is also making life harder for the parasite by trying to defend itself. So there's generally antagonism on both sides there. And so evolution can push these systems kind of in both directions along this continuum, towards mutualism, towards parasitism, etc. And it would be really nice if we could know when that's going to happen and how we can control and predict it. A couple examples of situations where this would be really useful are, so for one, phage therapy. So if someone is infected with bacteria that they don't want to be infected with, we can give them phage, which are viruses that infect bacteria. The phage can kill the bacteria by basically acting as a parasite or perhaps even a predator on the bacteria. Obviously, for this to work, we want to ensure that those phage are not going to evolve to be mutualistic with the bacteria. Predicting the outcome there is important. We also sometimes want the opposite. A lot of our plants that we grow in agriculture really benefit from having mutualistic interactions with soil microbes. And so if we could, again, predict when evolution is going to give us mutualism there, it would be useful. So what circumstances actually allow mutualism to evolve? When I talk about de novo evolution of mutualism here, what I mean is mutualism evolving in the context of antagonism. There's a whole line of research about what maintains mutualism once it exists, but here I'm asking about the origin of mutualism in the first place. And so one of the main hypotheses that's been put forward was from Baskett and Shemsky, and they thought that large effect size mutations are important. And I'll go to the next slide when I get a chance. But so the reason that they thought that they might be important is that if you imagine a parasite and an antagonistic host kind of locked in an arms race, it's probably deleterious to become friendlier for either of them. So there's potentially a large fitness value that needs to be crossed. And so Baskett and Shemsky were saying, maybe we need very large effect size mutations that can just jump straight over. It's broken. Okay. I apologize in advance. That's okay. Yes, so there's kind of two ways that this could work. So the one that I just described is kind of what I'm calling the instantaneous hypothesis, where these large effect size mutations just allow kind of teleportation directly from the parasitic state to the mutualistic state. And it was really interesting to me that that was kind of a leading theory, because when I think about the evolution of mutualism, I usually think about kind of, a complex co-evolutionary scenario where there's, feedback loops and interactions, et cetera, that eventually lead the whole system to a state. And so that's not what would happen under the instantaneous hypothesis, but it is an alternative possibility that maybe instead there's more complicated co-evolutionary dynamics and there's this gradual de-escalation where both parties move towards being friendlier to each other. So how can we test this? It is notoriously challenging to change the effect size of mutations in the lab or in nature. So this is a great use case for artificial life. I used a system called Symbulation. And so in Symbulation, there are two populations, the hosts and the parasites. It's designed to be very simple. So each one has a genome that is a single floating point value between -1 and 1. Resources are constantly flowing into the environment and the hosts receive them. If the host's interaction value is greater than 0, then that is going to indicate the proportion of its resources that it will give to the parasite. And similarly, if the, symbiont, it's probably not a parasite in this case. Similarly, if the symbiont has a positive interaction value, that will be the proportion of its resources that it will give back to the host after multiplying them by a synergy factor that models division of labor. So both parties can come out ahead here if they both cooperate with each other. However, they can also be antagonistic. So if the symbiont has an interaction value less than 0, that will be the proportion of resources it tries to steal from the host. And if the host has an interaction value less than 0, that will be the proportion of its resources that it invests into defending against the symbiont, making the symbiont's life more difficult. When organisms accumulate enough resource, they will reproduce. When they reproduce, a mutation will occur to their offspring. These mutations are drawn from a normal distribution centered at 0 with a width determined by a mutation size parameter. And there's also a mutation rate parameter that controls the probability that a mutation happens at all. Okay, so I'm actually going to skip over, I think, a lot of this math in the interest of time, but we can derive a simple mathematical model for when we would expect evolution to occur under the instantaneous hypothesis, or the evolution of mutualism to occur. Basically what it comes down to is, we can figure out the effect of a single mutation. Ooh, where is my normal distribution? There it is, okay. We can figure out the effect of a single mutation taking a host from its current state to being mutualistic. We can also figure out the probability of that happening to a symbiont. We can multiply them together to get the probability of a paired host and symbiont both having just gotten a mutation that makes them mutualistic to give us an actual mutualistic pair. We can subtract stuff from 1 and raise the powers to get eventually the probability of evolving mutualism. So if I had more time, I'd talk through this, but yeah, this equation should give us the probability that mutualism is going to evolve de novo under a given set of parameters. So we can plot this as a heat map. Here along the x-axis, we've got mutation rate. On the y-axis, we have mutation size. The color here is indicating the probability of evolving mutualism. And you can see it's a pretty steep transition between the region where we would expect mutualism to evolve and the region where we would expect it to not. There's a small little interstitial space where it might or might not. And so we can see if we see the same thing in Symbulation. So we conducted a set of experiments with 30 replicates per condition, with conditions sweeping various mutation sizes and mutation rates. And we started all of the interaction values here at -0.5. So they all started antagonistic. So this heat map shows the maximum symbiont interaction value occurred that we observed in each condition, each of the 30 replicates within each condition, and then those maxes are averaged together to give you the value shown in the C plot. Again, we have mutation rate on the x-axis, mutation size on the y-axis. Anything purple is going to indicate that mutualism is happening. Anything green is going to indicate that it's not. And so you can see that at first glance, this kind of looks like the same shape that we were predicting from our math. However, if we look a little closer and do a quantitative comparison, we can see that there's actually a larger region than we were expecting where we're seeing mutualism. And I did some parameter sensitivity analysis that showed that this was not just because of errors in our estimations of the parameters or anything. This seems like it's a real effect. Okay, so why is that happening? Well, okay, this is an overwhelming graph that I might skip in the interest of time. So we can look at the trajectories of how symbiont interaction values change over time. And basically what we see, in short, the thing that I kind of want you to take away from this is in those regions where we were expecting under the instantaneous hypothesis that we'd get evolution of mutualism, we see just immediate evolution of mutualism. The proportion of symbionts that are being mutualistic just always jumps right up. In that region where we saw mutualism, but we weren't expecting to, it happens a lot more gradually. We have, kind of different lines for different replicates that have mutualism occur at different points. So that's starting to look a little like the gradual hypothesis, but it's not clear if it's actually kind of like coevolution or if it just, takes longer to get the one mutation that makes you mutualistic. So to further answer this question, we can do a phylogenetic analysis. So these are some molar plots. On the left, we have hosts, and on the right, we have symbionts. So the way you read these is that each polygon represents what I'm going to call, for lack of a better term, a species. Feel free to talk to me later about the nuances there. The color of these indicates whether they're symbiotic or they're mutualistic or antagonistic. The greens and yellows are mutualistic. The blues and purples are antagonistic. The size of the polygon along the y-axis indicates the population size of that species. X-axis is time. And then when you see one polygon emanating from another, that is, one species giving rise to another species. And so we can see here is that we're getting kind of this progressive de-escalation where more friendly species are coming from slightly less friendly species. So this is looking a lot like gradual de-escalation. So we wanted to explore it a little more quantitatively. So we looked at the phylogenies from a sampling of our populations. This analysis turned out to be a little expensive, so we couldn't do it on all of them. But basically, so what we looked at here is we found the first mutualism that went on to be successful. So the first instance where there was a mutualistic host with a mutualistic symbiont, and they had, offspring eventually that were alive at the end of the run. And then we took those and we looked at their parents to see, to see how they evolved. So on the left here, we have a lower mutation size, 0.15, and we have three different mutation rates. And these colors indicate what the behavior types of their parents were. So these orange here are neutral, which means both the parent host and the parent symbiont were neutral. So they weren't antagonistic, they weren't mutualistic. This pink color shows that I think the symbiont was actually mutualistic. So at this lower mutation size, we're seeing a mix of ancestors that were neutral or actually mutualistic. At the higher mutation size, we see a lot of these dark purples, which indicate that both ancestors were antagonists. And so that shows that in that circumstance, we were indeed seeing that direct mutational jump from pure antagonism to pure mutualism. So what does this tell us overall? Well, first of all, it does seem like mutation size matters. So Basket and Chemsky were right about that. If the mutation size is too low, we don't get mutualism at all, even if we wait a super long time. And I'm still really curious to investigate the exact details of why that is. But if mutation rate is high enough, mutation size is high enough, there's kind of two regions of parameter space that behave a little bit differently. We have the zone where we just get this instantaneous mutation immediately to mutualism, but outside that zone, we still have a region where we can get this gradual de-escalation where coevolution allows the evolution of mutualism. So with that, I just want to thank my co-authors at my lab again, and thank you all for listening.